Oh, geez. Well, don't ever leave the screen is what I'm getting out of this. Cool. Here. Here we go. Ooh. Okay, sorry about that, guys. Like usual, we'll probably wait till 5 after, and then we'll get started. That's it. I guess the music stopped. So I'm sitting outside in my backyard, as I like to do when it's nice like this. I only have one tree in my yard. It's pretty big, but it's a magnolia tree. And I don't know if you know this, but they are the worst ever because they drop leaves year round and they have like weird stages of like seed pods that they'll drop. Yeah, I mean, they have nice, pretty flowers, but then those dry up and fall on you too. And right now, it's dropping these weird red seeds. I mean, I'm glad the weather's nice, but All right. I think that's enough time. Let's go ahead and get started. You guys know the drill with this. You don't need to see it. Good job. Good job. Good job. The part always stresses me out. Oh, it was close. I really thought you guys were making it. close. God. Oh, I really thought we were making it. It was a lot closer that time, though. Yay, made it. It's all right, we can do this. No. Alright, questions are a little bit more tricky than what we seem suggesting. That's why we're here. Alright, we'll do the next one. Next one we'll do one of the ones where we build the towers. Let's see. Tower. Remember, try to stack them up and get them in a row. I'm gonna let everybody join in. It should just automatically add those who are already playing. But if you want to join in, please do. Okay, so when we're since some of you might not have played this, we'll go over the little tutorial for it. You want to build the tallest tower with your team. Of course, you're going to get blocks if you answer questions, and you want to drop them. If you make them in a row, you build the whole floor. And the goal is to build 2 floors. Orient them. Sometimes it'll offer you bonuses. Disrupts everybody else. All right. Good luck, everybody. There we go, Team Finger. I was like waiting for you guys to drop it on that side. Uh-oh, monster. Okay, well, the music's on. I hate when it does that. Oh, the sound effects. I don't know if the music plays for you guys, but I'm sitting here in silence. Um, what was I gonna tell you? Oh, I was doing my laundry earlier, like a normal person would do, and my we bought this special fabric softener that's supposed to help with getting rid of odors because some of our towels, our newer towels, because they're so fuzzy, kind of hold in some of uh, the kind of, I don't know, musty smell that the towels will get if they hang out too long when they're wet. So we started washing with this. Anyways, I added it in to wash my bedding this morning and then set it aside, and it like slid off of the side of the top of the dryer and like fell behind the washer and the dryer, and it came open and the whole bottle just like poured everywhere behind the washer and the dryer. It's sticky and it smells like fresh fake apple. It's yeah, it's all right. Worse things can happen, but there's my story for the day. Oh, whoops, I didn't do that. Man, Team Kangaroo over there killing it. Getting close. There's a squirrel. Meatball. Alrighty. Guys did great. We will go on, I guess we'll go ahead. We want to do the chill ones yet. I don't know if we said the game before the chill and so the chill further. Meatball, that's the podcast. Let's see. What we got? Questions correctly to collect treasure for the trove. Tap the treasure as it flies by, but don't get too greedy. Let's try it. Yeah, because I don't know if this is a.
My daughter saw us playing this. She says she likes playing that one.
Oh yeah, the treasure trove one?
Yeah.
Okay, we might as well try it. I've never done it before. I don't know why. It's been there this whole time. Okay, just like a minute for if anybody else wants to join. Boy. All right, let's go ahead and get started. Just what first? Let's clarify. Only reflect the rules, not the junk, I guess. Here we go. There's so much junk over here on my friggin' deck. sounds like something crazy going on. But it is. I'm just passing through, sorry. Don't want the dogs barking. That just seemed to happen all of a sudden, but yeah, here we go. Yay! Right. Awesome work, guys. There it is. All right, now let's go ahead with the chill. I think I still have at least one. Of at least one that we haven't opened yet, I think. All right, let's go ahead. Go ahead and get started. I don't know. This one looks chill. Let's do this. It has water. It looks like it's gonna have water anyways. Hopefully that's water. my gosh. Squirrels are trying to kill me. Oh, that's Niagara Falls, isn't it? Oh, it's a Thomas Cole painting. My mom. The going over the in-class questions for answering. Usually I would call on you guys for it. It's pretty busy this semester, just a lot of info. I'm over that and stuff. So it happens. Let's still benefit from things that you might have seen. And look there. Let's see. Let's take a lot of my battery. Let's close out of some of this stuff. Ooh, that's probably good. Okay. All right, so if you guys have any questions along the way with this, let me know. I don't get any notification on like when I'm talking. If you guys were to ask questions on chat, like it doesn't pop up for me automatically. So if you guys see that somebody is doing that and you are somebody who is talking, then holler at me because I don't want to miss anybody's questions. Okay. So let's start out with chapter 6, our genetics chapter. So the first question would be, what are the 3 parts of a DNA nucleotide? And so I'm not expecting you guys necessarily to answer out on these. You're welcome to if you'd like. So I'll give a little pause for you guys to think of it, or look at your notes or whatever, but you don't have to answer out loud on these. All right. I'll go over the answers. Having said that, there's a little pause in and of its own. 3 parts of DNA nucleotide are going to be the nitrogenous base, which is what makes it A, T, C, or G. Then we have the sugar, which is a 5-carbon sugar. It's deoxyribose. And then we have a phosphate group. Next, we have explain what makes DNA replication semi-conservative. Let's see if I can pull up my writing utensil really quick while we're break time. There it is. Just in case. All right, so semi-conservative, because when you start off with your original sound effect apparently, and you separate them. Those are your original halves. Then the new ones will come in. So we have original and okay, well, apparently I'm gonna write backwards. Original. Anyways, and then new. And this one would be new. Didn't think that one through, but you get the idea. That's why it is semi-conservative, because we are only semi-conserving the original strand in the new strand, since the new one is half old and half new. Okay, so next we have during DNA replication, which strand makes the Okazaki fragments? The Okazaki fragments are generated by the lagging strand. Because DNA replication can only go in one direction, which is that 5' to 3' direction, as we open up our replication fork, we can go cranking on down with that 5' to 3' leading strand, but the other strand, as it is opened up gradually, can only be made starting from where the opening is. So we would have to do, it opens up more and more. So we make it in little pieces, and those pieces are called Okazaki fragments, and it's the lagging strand that makes them. Generally speaking, what is the overall purpose of transcription? All right, so we know DNA to RNA is transcription. If we're scribe, we're writing, in this case, writing in the same language, and transcription from DNA to RNA is to generate a working copy of the gene that you are wanting to make a product from. So you don't have to rely on taking the DNA out to work with it outside of the nucleus. We can make a copy of mRNA version in order to take that out of the nucleus and keep the DNA where it belongs. So what is the function of mRNA? So that is our working copy. It's kind of the same question, I guess. It's our working copy of that gene. All right, for the second part of chapter 6. This would have been the end of the class. All right, so what is a codon? A codon is a set of 3 nucleotides on the mRNA specifically. So codons are not on the DNA. Codons are on the mRNA, the copy of the DNA, the working copy. What are introns? I will say this, a codon as well. It's 3 nucleotide bit, if on the mRNA, that 3 nucleotide bit, that codon codes for an amino acid. So if you need an amino acid made, every amino acid that you call in to make your protein, each of them had a 3 nucleotide codon that called it in. There's like 5 wasps out here. All right, what are, they're all up in my tree that I hate. I hate my tree. All right, what are introns? Introns are pieces of, I say pieces, they're code in the gene. I'm not doing that. I'm out of here. All right, they are a sequence, like the genetic sequence within the gene, and therefore in the mRNA that you are transcribing, that doesn't code for anything useful. It's just nucleotides that don't mean anything for the product of the protein that you're trying to make. So because they're not useful, because they're useless, what we're going to do is go ahead and cut them out of our working mRNA copy so that we don't have to, so we can just read it on straight through. We don't have to worry about these parts that don't code for something. So that's the introns. And on our messenger RNA that we originally make, we will cut out our cells, we'll cut out the introns and join together the other pieces that actually code for things. The things that code for things, those pieces are called exons. What type of operon is usually on and will be turned off when enough product is made? So we were looking at this question in terms of like on a test or something like that, and there of course will be choices, but an operon that starts off as being on and then will end up being turned off, then what we're going to do to that operon will be repressing it, right? So ones that are already on, that we can turn off or repress, they're called repressible. Right. And usually the repressor, the thing that is signaling to turn off that repressible operon, is the product of the whole operon. It's just not getting used up by the cell enough, and so it will build up and end up acting as a repressor instead of being used up by the cell. What type of horizontal gene transfer involves competent cells taking up exogenous DNA? I would hope that you guys would know this one since we actually acted it out in the lab. This one is transformation. I don't really have anything else to say. I mean, that's just the definition of it. If a mutation causes AAA, which might code for lysine, to change to ACA, which might code for threonine, and these being amino acids. And then this being a codon, right? Just to remind everybody. We made one nucleotide change, and that nucleotide change resulted in lysine being turned into threonine. So it's a different amino acid. So anytime you have a change of an amino acid from a mutation, that's going to be called a missense mutation. All right, moving on to chapter 8. What type of sequence do restriction endonucleases cut at? So they are always going to recognize palindromic sequences. So in the case of palindrome in DNA, then what that sequence would potentially look like would be like ACC, GGT, and then the other way, if you're going at 5 prime, this was 3 prime direction. The other side has to be anti-parallel. So then that would be TGGCCA. So not only could you potentially fold these back onto themselves if you folded them right over at that middle, but if you read from A through to T, it's the same on the other strand. So that's how palindromes would work in this particular case, what we mean by that. How are RFLPs used in comparative and investigative science? There was a question like this in the review, Kahoot. So RFLPs are restriction fragment length polymorphisms. And what that means is that if you cut somebody's DNA with a restriction enzyme and you cut another person's DNA with a restriction enzyme, the fragments that you would get would differ between those people. And so you can use that to compare DNA samples, for example, to see if somebody was, a murderer. If you had a sample of their blood from the crime scene, you could also use it to compare differences in DNA sequence based on the fragment length that would arise. Wouldn't give you exact details on the difference in the sequence, but it could give an indication that there was a difference. All right, so what does PCR do? It amplifies DNA. We're going to make copies of DNA. That's all it does. It does nothing else. Okay. It doesn't diagnose. Nope. It does not diagnose. It only makes copies of DNA. What are the 3 basic steps of PCR? We have first denaturation, and that's done by heat. Separates the strands. The second one is anneal. So we're doing that with the primers. To cool it down a little bit in order to get those to be able to bind, because if it stayed the same temperature, the same heat that separated the original strands will still separate the primers from the strands. So you have to cool it down a little bit for that. And they have to be specific enough that they're not going to bind at just any old sequence in a DNA sample, that they want to bind at a target sequence, the one that you know is there or should be there, and you're looking to see and confirm that it is there. So that primers will help with that. Number 3, this is going to be elongation. So that's just adding nucleotides, the complementary nucleotides. So A to T and G to C, etc. And you repeat. All right, in gel electrophoresis, which pole do DNA fragments move toward? Well, DNA is negatively charged because of the phosphate groups in the backbone. So the DNA will move toward the positive end. I say I'm giving like you guys some pause to answer it, but I guess I lied. So I apologize for that. You guys are reviewing these are available to you guys on the announcement for the Unit 2 review. And you can find these slides, a link to the Kahoot. I made it an assignment so you couldn't like click through it like you would normal practice Kahoots. This is available. I'm trying to think what else. There's a couple of things on there that might be worth looking at if you want more review resources. Anyways, which one of the following is the largest? 2 kbp, 3000 bp, or 55 bp? So it's going to be this. What? I don't want to exit. Whatever. So what we would pick would be this one, because it's 3000 base pairs, because this one, when you write it out, and you're welcome to do that on your scratch paper, would be 2000 base pairs. So that's why that one would be the answer. Why can restriction fragment length polymorphisms be used to tell the killer from the innocent suspect? I already explained this one, so I'm not going to go back over this one. It's the exact same as what I was just saying. You cut different people's DNA, you're going to get different lengths of fragments because everybody's DNA sequences are different. Oh my gosh, Spaghetti, you're so cute right now. But how can a SNP be so dangerous? So an SNP is a single nucleotide polymorphism. That means you have one nucleotide change. We saw previously, we were talking about lysine to threonine. You can even have a single nucleotide change a hydrophobic amino acid to a charged one, or vice versa. You can imagine the effect that could have on a protein's structure. And remember, protein's function is very highly dependent on its structure. So that could ruin the entire function of a protein. And if you need that protein to live, it could be deadly. All right, what is a GMO? A GMO is a genetically modified organism, aka a transgenic organism. It means that you took genetic information from one organism and put it into the genetic information of another. So now, of course, there's some. I think there's, sorry. If a method of gene therapy affects somatic cells, can the treatment be passed to future progeny? The answer for this one is no, because somatic cells are going to be your adult tissue cells. It doesn't affect like gametes, like your sperm or your egg. You would need to be dealing with germline therapy for that. What? Okay, chapter 9. What is a chemoheterotroph? A chemoheterotroph. This is two things that we can get out of here. We can get where the carbon is coming from as well as the energy. So carbon source, we get that from the heterotroph part. And then the energy source, we get that from the chemo. In this case, heterotrophs, which, hetero being different, heterotrophs are getting their carbon from a variety of different organic molecule sources. They've already been, we're not talking about inorganic there. Hetero is going to be talking about the organic. They're already organic. How did they become organic? Are they just naturally existing that way? No. Another organism had to turn inorganic carbon into organic carbon. And then we are getting that organic carbon from them. So of course, them being autotrophs, because they can auto make their own organic molecules. All right, so our chemo is referring to their energy source. This is energy from breaking chemical bonds. The other option, of course, would be phototrophs. Phototrophs get their energy from the sun. So kind of the opposite or a parallel for chemoheterotroph would be photoautotroph for these. Okay. What are saprobes? Saprobes are organisms that break down dead or decaying matter. What would happen to a cell if you put it in pure water? If it's pure water, that means there are no solutes at all in it. We use a lot of the time in our examples to talk about solutes, we talk about salt, but that is not the only solute that there is. Anything that is dissolved into water is a solute, anything. And so if we're pure water, we would say there's no solutes in that water, right? Does the inside of your cell have any solutes in it on its own? Yeah, it has a lot. Even nucleotides are going to count here. Nucleotides that might be in your cell, the proteins that are in your cell, the amino acids that make up those proteins, salts and other sorts of metal ions that might be important for the function of proteins in there. All of these things are solutes. So all of this is hanging out in the watery makeup, that cytoplasm inside of your cell. And cytoplasm straight up isn't just pure water either. So if we're putting a cell into pure water, the way that physics would want us to work would be that the water would go into the cell to try to dilute out the contents of the cell so that the concentrations of solute were the same inside the cell as they are outside of the cell, which we can already see is a losing battle. There's impossible. There's no way you could take up all of enough water to water down solutes in the cell, because you can't get rid of the solutes in the cell. They're still there. So you can't ever get it. Anyways, that's a battle that will be lost by the cell. And what happens is, could potentially, as the cell swells up, trying to take up the water to compensate for that, could burst. It could lyse. All right, why does active transport require energy? Well, just like it says, an active transport, if you're doing an activity, you need energy in order to do the activity. Active transport usually is a transport mechanism that is going either against a gradient. So there's very little of this outside of the cell, and there's plenty of it inside the cell, but we want more inside of the cell. Then active transport can do that for us. If we feel like the cell isn't bringing in, even if you had, like, let's say, plenty of stuff outside of the cell and could use diffusion to get it in, if it's not coming in fast enough and you want it to come in faster, then you can use energy to help that process. And that's also active transport. So anything where you're going against what would naturally happen through diffusion, then you would call that active transport generally. And it will always require energy to accomplish that. All right, a mesophile, one of the things that we would have gone over in class if we'd had time. A mesophile, these are organisms that live at middle temperatures. So if you live here in this, where we are right now, where you're sitting right now, you and any organism inside of you or near you, all of those are mesophiles. We have thermophiles that will live at high temperatures and can even flourish at temperatures above boiling. So those are the hyperthermophiles. We have psychrophiles, which prefer to grow in cold, even freezing conditions. But the mesophiles, they grow at middle temperatures. All right, chapter 25. 88% of the ocean surface is covered with what? And this one, if you guys listen to the lecture, 85% of the ocean's surface is covered with plastics, microplastics to be specific. They can join together and create these big sort of rafts that can, alter how much sun gets to lower levels, create an area for different microbes to grow there that shouldn't be there, that can affect the whole entire biome in general. So this is a horrible thing when we say there are islands of these plastics. Like one of them, I was reading up on this, one of them is the size of Texas. That's ridiculous. What happens to reclaimed wastewater? As I'm sure we were all paying attention from the presentations from our peers, reclaimed wastewater can be used to water parks, parkland, or water golf courses, or it can be returned back into the water system. So including rivers and lakes and things like that. What is the term for water that is free of pathogens, toxins, turbidity, odor, or taste? There's nothing in it that's going to make you sick or make you not want to drink it. This type of water, there's a term for it, it's called potable. There used to be a way, like a saying or something that I had to help you guys remember that it was potable and not potable, but I forget it now, unfortunately. I'm sure I'll think of it at some point. I'm just really tired right now. Potable, I don't know. All right, what are coliforms? Coliforms are essentially going to serve as indicator bacteria in our water supply or other, say water supply, but it could even be like food or something that is being prepared essentially. And that when that is being prepared, we want to test it to see if these coliforms were present. Now, coliforms aren't the thing that's going to hurt you. The coliforms are there to say, if these guys are indicator guys, if they made it through this, pathogens could make it through. Does that mean that pathogens are making it through? No. But if our normal E. coli makes it through, then perhaps Salmonella or Shigella could make it through, or cholera. And before they make it through, we would like to know if it was possible to make it through. And that's what these guys do for us. That's what coliforms do. And they are typically fecal matter coliforms, either fecal coliforms for people or for wildlife. And they should have, none of that should be making it through the purification process. Oh my God, they're running with a toy. Okay, what process is alcohol made by? Okay, I read it weird. By what process is alcohol made in beer? So this is going to be talking about fermentation. And this chapter would have touched on, how the uses of organisms, and it didn't quite get into fermentation itself, which was in chapter 10, as I'm sure we are all aware. But beer is manufactured via fermentation. So the alcohol and the carbon dioxide that we find in beer are both produced by organisms undergoing fermentation. All right, going on chapter 10's questions. What is catabolism? Remember, cats are going to tear things apart, break things apart. It's just like the bonds. So catabolism is where you have breaking bonds, and that will release energy. It is one of the subsets of metabolism. So meta, right? Meta, it means all of it. Catabolism is the breaking down, anabolism is the building. What is the term for the molecules that enzymes act upon? The thing that they are helping go through the reaction is called a substrate. What is oxidation? Well, Leo says GER or oil rig. And so oxidation would be loss of electrons. Anytime that electrons are lost. Come on. All right, explain competitive inhibition. So competitive inhibition involves the inhibitor, whatever it is, binding at the site where the substrate would usually go. That site's called the active site, right? So if we're binding as an inhibitor into that active site, it can become quite a tight bond and can last for quite a while. So inhibiting the ability of that enzyme to work for a while. So that's how competitive inhibitors will work, by competing with, against, I would say, the inhibitors are competing against the substrate for that active site, and they usually would win. How does ATP store energy? Well, this is just referring to the potential energy that is made as a result of having those phosphates that are very, very negatively charged and repelling one another. We have covalently bonded them together, forced them together. So they don't want to be anywhere near each other. So they're constantly pushing each other apart. And when you break that bond from that phosphate to the one next to it, you can use that potential energy, release it to drive processes in the cell. I thought that wasn't enough. Was that all of that for that? Yes, it was. Okay, I don't know why it didn't seem like enough. All right, part 2. What are the 3 steps in aerobic respiration? This is a big idea steps. So that's going to be glycolysis. The next one, Krebs. And the last one, the electron transport chain. What is the major purpose of glycolysis? To generate pyruvate, right? Pyruvic acid, which is a metabolic intermediate for making a lot of molecules in the cell, as well as a necessary molecule for leading into the Krebs cycle, essentially. So pyruvate. All right, when you exhale, where did those carbon and oxygen molecules that make up the carbon dioxide originate from? Those are going to have originated from the glucose molecule that you put into glycolysis and cycled its pyruvate into acetyl-CoA and then Krebs cycle, and going from that through that whole process releases carbon dioxide as we steal electrons and kick off carbon as a waste product. So it came from the food. Explain why it's important to keep breathing. So we've talked about this up and down. You need oxygen to help maintain that proton motive force, that gradient of the protons outside of the cell. We can't let them build up inside of the cell, or there's no more gradient to drive ATP production. Additionally, in order to move electrons through the electron transport chain proteins, those cytochromes, and keep exciting them to allow us to make that proton motive force, right, through chemiosmosis. So in order to keep that going, we have to get rid of the electrons before it. So we have to get rid of both of those. So that's why we need oxygen, so we can take those leftover H pluses with the electrons, make them into just H's to share their electrons with an oxygen atom, and we create water as a byproduct from doing that. So oxygen is our final electron acceptor because of that. All right, by which pathway or set of pathways do plants get the majority of their ATP from? Hate to break it to you, aerobic respiration. I feel like at this point, if you need me to explain that, maybe you weren't paying attention to the lectures. But yes, for photosynthesis, we make only a little bit of ATP to power fixing inorganic carbon to make 6-carbon sugar molecules like glucose. Those can be shuttled through aerobic respiration to make most of the ATP for the plant. Anytime you need a lot of ATP like that, you're going to have to make glucose or whatever. So, anyways. Chapter 13, define disease. Disease is any deviation from health, whether it is due to an infection or any other thing going on in the body, doesn't matter. If you're deviating from health, that is disease. What is an endogenous infection? An endogenous infection is an infection that stemmed from microbes that were part of your biome already. So you already had these microbes in your body, and they either moved somewhere that they didn't belong, like the urinary tract infection with the E. coli, or they overgrew in the area where they were, like the yeast in the yeast infection example. All right, what is a virulence factor? Well, remember, virulence is just a degree, and it says in the notes and everything, it's a degree of pathogenicity. Pathogenicity is essentially presentation of disease. So you've got disease going on, and virulence is basically a measure of how bad it is. A virulence factor is any attribute of an organism that would make it virulent, make it show the disease more, and to establish the infection. So things like capsules, which help get us evade phagocytosis, or flagella that help us move around in our environment. Anything that helps the organism establish that disease, that is a virulence factor. Is a headache a sign or a symptom? Can you measure a headache? Can you see the actual headache? Can you touch the actual headache? No? And it is a symptom. Can you see, if we look at the word sign, like I said before, if you saw on the side of the road a sign of any kind, a stop sign, a yield sign, you can see, you can touch. So you can't, there's no iteration you can give me of symptom that would work that way. Yeah, so that's how I remember that sign is the touch and interact with, measure one, and symptom is the subjective. So it's reported, but you can't verify it. Not the same way, you can't measure it. During which phase of infection do the very first signs or symptoms first start to appear? This was one of the questions on the review, if you got to it. This is the prodrome or the prodromal phase or stage. All right, chapter 14, this is dealing with epidemiology. So first we have who was responsible for identifying and eliminating the cholera outbreak in the 1800s in London. So the guy that did that, his name was John Snow. I don't know, that's his name. That's who it was. All right, what is the difference between prevalence and incidence? A big one for a lot of people that will get this one wrong. I feel like there's a question on this one. It might have been on the final, but I'm almost certain it's on this exam that you're actually going to be asked a question that will require you to know the difference. So prevalence is looking at the total cases in the outbreak and looking at that outbreak, okay? And whereas the incidence is looking at the number per time period. So looking at weekly cases of COVID, looking at morbidity report for COVID. Remember, the reason I'm saying morbidity is because incidence and morbidity are interchangeable. They mean the same thing. All right, endemic, that's a term referring to something that you expect to be in that location at like that level that it's at. So prior to COVID, think back like 10 years ago. What was the expected level of the COVID-19 virus, that SARS-CoV-2 virus? Zero. It didn't, it was, no, it wasn't here, right? It didn't exist 10 years ago. So that would have been zero. That was the rate that you would have had. Now, that's not necessarily the case. Now you would expect to see a few cases. It's not like it was when it first came along. I don't know what the exact, number you would expect to see every week, but you do expect to see it. So it's become endemic, and any deviation outside of that, like an increase from what you were expecting, would be an epidemic. Right, what is the difference between sensitivity and specificity? This is hard for me to explain. All right, when you are sensitive, you will find every single positive, if there's one. And I mean all of them. If it's super, super, super weak, on a test, like a COVID home test or something, the more sensitive it is, the more it's going to notice that it's even just a little bit positive. Okay, so sensitivity, moving towards finding all positives. And when you do that, this means you should be moving towards less false negatives. What's a false negative? Saying something is negative when it's actually positive. It's a false negative. It's falsely called negative. Okay. So you can see if you're really, really sensitive, you shouldn't have anything where you're saying something is negative when it was actually positive. You just didn't catch it. Right. That's what sensitivity is describing. So specificity is different. It's completely different. It is not dependent on the other one. They are not, mutually exclusive. No, you want both of these. So specificity, you will find only positives. And what I'm saying here then would be like you will decrease or have less false positives. Because a false positive is saying, of course, that it was positive when it's actually negative. So you shouldn't be calling something that's negative positive ever. So we're very specifically only interacting with positives. So you want both, obviously. You're going to only interact with positives, and you can find any of the positives, even the super, super, super weak ones. That would be highly sensitive and highly specific. All right, herd immunity. This is something that would occur if you, depending on the, how contagious a disease is, how communicable it is. There is a certain number of people in the community that can be vaccinated, that if they reach that threshold in that community, they are protecting the ones that aren't or can't be vaccinated. That is to say that because this disease is not super contagious, as long as we have 75% of the population vaccinated, it's not likely to get within the population enough to infect the other 25%. That's an example. Whereas we talk about something like measles, which is very, very highly communicable, one of the most communicable diseases ever. And you would need something like 96% of the population to be vaccinated in order to protect the other 4% that can't be vaccinated or aren't vaccinated, which is why It's the first one that you see pop up with the anti-vax movement. Okay, so that is it for our class questions. Oh man, I keep picking these rooms in the house that are like I have to keep moving. Either wasps or now it's the sun literally beating down onto my body. Let's go to the office. That's probably where I should have been to begin with.
So my lights just turned out. I got lights all around.
Okay. Let me see. Hold on real quick before I go into these next ones. Okay. I just want to see how many people we've got still hanging on. I'm still 9. That's not so bad. If I lost all you guys along the way, I would have just been like, oh, guess I'm going, I'm done. All right. So these, I called this one questions along the way. These are meant to be questions to kind of think about while you are reviewing your material. If you're starting to feel pretty comfortable with your material and now you want to challenge yourself because the knowledge part is out of the way, but now the understanding part is something that you want to work on, then these are meant to be questions that you would think about while you are kind of going over it that way. Some of them are just basic questions, but some kind of apply a little bit more thinking about them. So first of all, we have where does replication take place in bacteria versus eukaryotes? We talk a lot about, we've mentioned several times about where transcription would be happening or translation or where aerobic respiration would be happening. But we didn't really talk about where replication takes place in bacteria versus eukaryotes. And we shouldn't have to because the answer is already there. Because eukaryotes, replication, which is always talking about DNA replication, DNA, we make copies of our DNA in the nucleus. So there we go for eukaryotes. But what about bacteria? Right? They don't have a nucleus. We should know that for sure by now. So their replication process, cytoplasm. That's all they have. They don't have anything else to separate cytoplasm from anything else. Everything that isn't along a membrane, like their inner membrane, like we talk about electron transport chain in bacteria being along their inner membrane, right? If it's not along that inner membrane, it's in the cytoplasm. There's nowhere else for it to be taking place. All right, why are Okazaki fragments made? So we know that they are made, and we know that they're made by the lagging strand. Hi, Spaghetti. Cute little boy. Yeah, you're a little noodle. Okazaki fragments are made because as we can only synthesize the new strand, the leading strand, in 5' to 3' direction. As we push into that replication fork, we are opening it up further. Whereas the lagging strand across from it, well, we're opening it up more and more on the end where we would have liked to have started. So we have to make it in parts. And that's why Okazaki fragments are made. Is facilitated diffusion truly energy free? Facilitated diffusion is just diffusion. We have a protein channel, perhaps, that things can diffuse naturally through. And the diffusion itself is energy free, right? It's depending on diffusion. It's just diffusion. But you had to put energy into making that protein channel. Protein channels don't spontaneously generate, right? So that's really what I'm getting at with this one. All right, is transcription considered an anabolic, catabolic, or none of the above? So I've taken 2 very different concepts from 2 very different chapters. Here I'm taking chapter 6, and I'm taking chapter 10. Believe me, this is not the last time that I do it. Transcription, what is it exactly? Well, we've already said it's making the mRNA. So we've had to make mRNA by linking together RNA nucleotides. When you link together RNA nucleotides, to make the mRNA complementary to your, DNA template, you're making bonds. That's anabolism. All right. Requires energy, right? All right. Why does transcription take place in the nucleus in eukaryotes? Makes sense. We just said with replication has to take place in the nucleus with eukaryotes because DNA, making DNA, of course, that's going to be in the nucleus. Why can't transcription take place in the outside of the nucleus? Has to be in the nucleus because we're using the DNA as a template. The DNA never leaves the nucleus, never leaves it. Okay, so that's why we're using it as our template. You are so cute, my guy. Anyways, all right, there's that. Okay, why does fermentation generate alcohol, carbon dioxide, or acid? This isn't a true or false, does it do this? Or fermentation, what products does it make? No. Why? The reason that we make alcohol or acid or carbon dioxide in fermentation is because when we have gone through glycolysis, which is the only process involved in fermentation, we have pyruvate at the end, as well as we make a little bit of ATP. That's the whole point for fermentation, right? But along the way, we have also made NADH. We have reduced NAD to make NADH, which if we reduced NAD to make NADH, we have what involved there? It's carrying what? An electron. And so you need to do something with it. So to handle the electron, we'll just say that, to take care of that situation, we put that electron right back in to pyruvate, our other product. It's what we've got. And how do we do that? I don't need you to know the chemistry of it, but involves breaking and or making bonds to accommodate that electron. And as we break or make bonds, shift around bonds to put that electron back into pyruvate, we create alcohol and or acid and or carbon dioxide. All right, why is it important to stay adequately hydrated? You need to stay adequately hydrated for a lot of reasons. But one of these, the homeostasis aspect, so making sure that everything's running smoothly, is we want to maintain your cells in an isotonic environment, right? So you keep hydrated so that your body can maintain that isotonic environment and therefore the happy environment for your cells as best as it can. And this helps out greatly with your filtering organs, your kidneys. How is oxygen serving a purpose in aerobic respiration? I mean, I've gone over this one as well. This is your final electron acceptor. It's taking the electrons plus the H pluses, turning them into regular H's. They can share their electron with the oxygen now, just like oxygen loves to hog those electrons from those H's in the form of water. Which epidemiological studies rely on at least some of the subjects having signs and symptoms from, let's say, day one of the study, okay? When we look at the cohort study, that's the one where you would say, these guys are smokers, these guys are not smokers, and we're gonna follow them over time to see if they get the disease. And then, look at the differences. Those are the ones that didn't have the disease, and we're seeing to see if they get them. So it requires that. The other ones usually involve having the disease during, like, at the time you're starting the study, usually. Let's see. How are correlation and causation different? Just because you notice that these factors align with one another or happen at the same time as one another or seem to end up in the same way as one another. It doesn't matter unless you can prove the causation. So correlating is just saying that they have these similar things going on that suggest blah, blah, blah. Causation is proving it. So seeing somebody standing over the dead body with blood on their hands, that's correlation. Proving that they actually killed the person, now we've got causation, and we've maintained the correlation. All right, which is better, sensitivity or specificity? I just went over that one. I didn't write these with the other questions in mind, so I didn't really think that through that way, but so I'm not going to go over that one again. Can you tell the difference between a complete protein produced by bacteria versus that same exact protein produced by eukaryotes? Why or why not? Okay, no, you can't tell. You can't tell. The processes in the middle of how it happens and where it happens in the cell and those things can differ, but the protein is the same in the end, no matter what. Even if there were introns, you don't see that in the protein, right? Proteins will be the same no matter what. Do cofactors or coenzymes bind the enzyme's active site? Never. Okay, that's where the substrate goes. The active site is where the substrate goes. Couldn't they be butted up to it and help with things? Yes. But are they in the actual true active site? No. The whole reason it's called the active site and does what it does is because the substrate binds there, not something else. So coenzymes and cofactors, they will involve themselves with the protein portion of the enzyme. So the apoenzyme. Remember how I told you guys how, well, I told you, like, I guess the notes, the lecture told you guys that conjugated enzymes have parts, right? And the holoenzyme is the whole working one. The apoenzyme is the protein portion. So I'm telling you about like the cofactors and the coenzymes and where they're going to be binding onto the apoenzyme. But where's the other part? I don't know what it is, but people always think like, okay, well, you said that there was other part, like the apoenzyme and what? That is the coenzyme or the cofactor. That is it. Like, that's exactly it. So it is binding onto that protein portion, the apoenzyme. And only when they're bound together are they a whole working enzyme. Okay? That way you have a functional active site for the substrate. Can eukaryotic genes, can eukaryotic genes ever be repressed or induced? Do not think that because we learned about operons, that those are the only things that are repressible or inducible. Operons are only a set of genes, more than one gene, linked together to serve a similar purpose that are all regulated at once in one place. Okay? So it's the operator that controls turning on or off the whole operon via using the repressor protein. And whether it starts off bound on there or can later become bound on there determines whether it is inducible, the former, or repressible, the latter. But our genes can be repressed, they can be induced. We just don't have them linked together reacting as a whole set of a bunch of genes to one operator area on our genes. They each individually have to be controlled. How do photosynthetic organisms harness light energy to generate ATP with light-dependent reactions? So we're going through photosynthesis in a light-dependent reaction. And what's going on is the light in the form of a photon that has its own energy just because it's moving will come in and excite, I want to say pigment molecules that are effectively the exact same thing as cytochromes in our electron transport chain. These pigment molecules become excited by the photon. they will release their own electrons involved from that to basically drive what is essentially the same thing as the electron transport chain to make ATP from that. All right, so what is an operon? Oh, like I already said this one, so I'm not going to say it again. You know, set of genes that are all going to be controlled by that one operator area. Oh, this was for me to take notes if I needed to. All right, so next we have the bringing concepts together questions. Here we're going to get meta. So if you're not ready for these, you might not be ready for these, okay? If you've been studying and you're working towards the understanding and not just the learning the terms and things like that, so you want to actually start applying it as opposed to just knowing it, that's really, this is it. You'll know when you get to these. Okay. How does aerobic respiration relate to the process of replication? You see what I mean about meta? Okay. So how do these two relate to one another? In so many ways. It's almost like a circle, you could say, but it's just these questions, I'm not sure I can necessarily even break them down to their complete entirety. But just to touch on this, aerobic respiration, what's the point of it? To make ATP, right? When you're replicating DNA, making new DNA strands, let's hope that you understand we're creating, right? We're making bonds. And that means energy. So there's that right there already. You had to go through aerobic respiration to make the energy to drive ATP, to drive replication. So there's that, but that's not it. That's not everything. When you go through aerobic respiration, most of the steps where we are converting these molecules into their different forms along the way, they're not just happening spontaneously. There are enzymes driving them, right? Every step. The ones that we aren't talking about, the ones in the background, they each have an enzyme driving it. Enzymes, last I checked, are proteins. And proteins, last I checked, are coded for by your genes. And those genes, last I checked, are part of your DNA. And that DNA had to be replicated to make that specific cell. So it's like a circle. You see what I mean? And you can relate it in a lot of different ways, and not just that one. And I challenge you guys to think your way through some of these and see what you can get as far as understanding these processes enough. These make sense. The next one says, how might the 4 major types of the 6 enzyme classes lead to the development of signs and symptoms? Whew, all right. So I told you guys, you don't need to know all 6 of them, and I'm not worried about all 6 of them, but the 4 that I was worried about, and the reason I left space out to the side is for this. Okay. I want to start this. Let's just list them first. Let's list the guys. Well, I wasn't expecting. Our enzymes are going to be oxidoreductases. There'll be hydrolases. We'll just say isomerases first. Or did we leave that one out? Transferase is what we went with, right? Pretend like that's not there. Hydrolases. And ligases. And these two were almost essentially opposites of one another. I could go through this and like try to break down kind of how these would be used at every single step of the way. But that's gonna take forever. So let's just look at signs and symptoms, right? So signs and symptoms, just basically what you see from the disease, what the disease is presenting, right? So we're gonna say just disease symptoms. I know we didn't say signs, but just for now, let's make it easy. you want to put signs, it's fine. Whatever. All right, so we're looking at how those cause that. When you are having signs and symptoms, typically they are usually caused by inflammation somewhere in your body. Sometimes it's local in one area, or it can be in larger areas, or it can be systemic. It just depends on what's going on in the body. One, like for example, if we look at one sign, fever. When you have a fever, your body had to use a lot of energy to make that fever, right? I mean, it doesn't just happen. So to give off heat, that's energy. So you had to make energy to do that. So we already know, if you need energy, you need ATP. And most of ATP's reactions are going to be driven by oxidoreductases because they're redox reactions, the transfer of electrons. Transferases are important for transferring groups from one molecule to another. There are several instances of how transferases are important in your body, but there are definitely transferases involved in the entire aerobic respiration process. I'm not going to go over why or how, but that is true. The hydrolases, these guys are involved in breaking down larger molecules into smaller ones. You haven't eaten in a while or whatever, and you start to, you're feeling sick or nauseous, and you start to like kind of waste away. When you don't want to eat, when you're sick and you just do not feel like eating, that's called anorexia. That is the word for it. When you have anorexia, your body will use up your fat stores in order to get energy instead. You need hydrolases to break that down. So weight loss from an illness or disease, you need hydrolases. Ligases, you are fighting off a disease and you need your immune system to build up cells to create antibodies and things like that to fight off disease. When you start activating your B cells or your T cells that are in your acquired immunity, they don't come automatically already in hordes. You have to select the only single right cell, the only one, there's only one of them, and then activate that, and then it proliferates. It clones itself. When that happens, you're making copies of the DNA. And already we talked about how replication process comes back to aerobic respiration again. We need DNA ligase to seal all these up. So all we have given an example of every single one of these four enzyme classes playing a part in any of this. Because you need, we say, for signs and symptoms, last time I checked, when you have an infection and you get your blood drawn, what's probably going to go up as a result? Your white blood cell count. Last time I checked, that's a sign. All right, we getting it now? You guys starting to feel where I'm coming from with this? Okay, I might have to bring down an eraser on this because I can't keep drawing over here. Like, what happened? Okay, I was like, what happened? All right, let's get rid of this. Get ready for the next thing. All right, how might methods, yes, that's where we are, right? We already did these ones. We did this one, and this one. How might methods of transmission relate to R naught? This one's not so bad, okay? And how might R naught relate to herd immunity? Okay, so I did mention to you guys that the more communicable a disease is, the more contagious it is, the more people need to be vaccinated to achieve herd immunity, to protect people that aren't vaccinated. So what's going on there is that R naught is very high. The number of people, remember this is what this is, R naught equals the number of non-infected people that you'll infect during the course of your entire infection. If something is very, very contagious, you're more likely to infect a lot of people than something that isn't particularly contagious or, gonna be exposed to just naturally. Like if you had, like me, when I had my sinus infection with the mold, I had aspergillus mold growing in my sinuses. Now, what's the R naught of having mold in your sinuses? If it's not 0, I'd be surprised, right? So this is what we're talking about. I was probably not going to infect anybody. If we had a vaccine for aspergillus, which I think they do, but if we had a vaccine for aspergillus, how many people do you think would have to be vaccinated against aspergillus in order to protect the whole population? They don't care because it's not a concern, right? But say it again with COVID, right? It is infectious and you are going to probably get people sick. In fact, that's what R naught is based on. People who are getting other people sick. On average, this is what's going to happen. So we have to have a certain number of people vaccinated against COVID so that the people who cannot get vaccinated are going to be less likely to encounter that disease from the people in their life. That's what herd immunity is about. Okay? Since R naught has to deal with the number of people you're going to infect with that disease, R naught is essentially also referring to how contagious. And that is really what herd immunity is going to be depending on. So we've done this one. We're looking at transmission. Basically, how does transmission relate to R naught? If like with the measles and how contagious it is, if it's super airborne, you sneeze, you cough, and it stays in the air really, really easily from that direct transmission. If it is transmitted from indirect transmission, like from fomites, like the pens that you're holding, or the table that you're sitting at, or the chair that you pulled out, or the door handle that you touched, if that pathogen is stable in those areas and transmitted in high enough numbers to those areas, direct to your next person, your next victim, or indirectly to the next victim, that would be a high transmission rate if it's able to do all of those things, let alone the infectious dose, how many of those organisms you need to get in your body to lead to the infection and therefore disease. So now we're talking about all of these things relating to one another, and essentially, of course, R naught being contagiousness. So the more able you are to be transmitted via these various transmission routes, and if you're talking about super contagious, probably all of them, the higher the R naught's going to be. The higher the R naught, the more contagious, the more people that are going to need to be vaccinated to protect the others that aren't, that they're going to encounter in their life. Makes sense? So these are more conceptual here. These are questions, again, that are going to be very difficult for people who have to have right answers or think that there is a right answer to every single thing. This is not that, okay? Are there wrong answers? Of course. But some of these, the right answer is vague. It's just about understanding and demonstrating that. All right, next. Explain how aerobic respiration relates to actual aerobics. I did this one in the study session. So if y'all want to know about this one, you can go watch that. Okay. All right. I highly recommend that you do, because I walk through with drawing everything out, the entire thing, and I talk through things with people. So if you weren't able to attend it live, please go and watch it and watch along how the progression of the whiteboard as we move through the topics. All right, and the whiteboard itself is available as well. Explain the processes leading up to replication, which are needed for it to occur. This is a very complicated one. And I give hints down here, which if you are like having a hard time even thinking where to start, you might consider how you have to transcribe various RNAs. Maybe you're already looking at this like, okay. So what is replication, right? We're making DNA copies. How do we make this happen? What do we need in order for that to happen, right? We need nucleotides, obviously. The A, the T, the C, the G. Nucleotides. And if we can't get nucleotides directly, then what would we need? We would need the bases, the sugar, or the phosphate. All right. What else do we need besides that? We will need the various enzymes involved. If you're a eukaryote, you need something called helicase. That would unwind the DNA. We also need the obvious one, DNA polymerase. And another obvious one, DNA ligase, right? There's a whole lot of other ones involved here that I'm not even going to touch right now. But we definitely should know that we need these for replication, right? Okay, so when you need enzymes like these, then you originally had to have the gene for that enzyme, which is a DNA, right? To get made into the RNA, to get made into that protein, right? Okay. In this process here, you don't just need mRNA, do you? Don't you also need tRNA? Don't you also need the amino acids associated with it? And don't you also need rRNA, ribosomal RNA? So this is just getting that first hint set up. It's not even going through the actual details of it, right? DNA polymerase is an enzyme. It's a protein that had to be translated. If we're building new DNA, we also need energy because we're going to be making bonds. So now we need everything that's involved in aerobic respiration and all of the proteins made that are involved in aerobic respiration. And we're going to need energy for that too. We need energy to start off with, just to make more energy. You can see how this is just the tip of the iceberg on how interconnected these things are with one another. If you go back and you go back and you go back, I'm making you write an essay about it. Think about how they're related though. And I feel like answering the questions on the test will be a lot easier if you're able to understand how they're related in a way that you can think back and it makes sense with all we've learned. So that's all I'm going to say for now. That one is a complicated one. There's way more that you can touch on, and I highly encourage you guys to do that. Don't just always rely on me to give you answers. It's not actually that useful if I give you the answer. All right, the next thing, explain events going on in aerobic respiration along with other cell processes that lead to aerobes dying within minutes when suffocating. So what this is asking us to do is say, okay, we know we need, this isn't asking you to say, to recognize that we need oxygen. This is not that. Okay, this is asking you to take the steps back from that. So if you're being suffocated, right, you don't have oxygen there. This is the basics, okay? I can draw this out in details, but it's just, I feel like you guys, it would benefit you more if you do the details on your own. But here's a walkthrough on the background of it. If you need oxygen and you're not getting it anymore, what will happen is you'll get a buildup of the H pluses and the electrons, right? Easy. And that would lead to us not being able to make ATP, right? Easy. Okay. I'm not asking you to stop there. Why is this a problem? So now we're talking about having to look at all of the cell processes, whether it is, and here's the thing, anytime that you need to do anything in the cell, ever, you need energy at some point. You need to move things around, you need energy. If you needed to make a new organelle, you needed energy for that. If you needed to make proteins to build new things, you need energy to drive the transcription. You need energy for the translation. You needed energy to make the mRNA. You needed energy to make the ribosomal RNA. You needed energy to make the tRNA. You needed, okay. So, so yes. So why is that a big deal? That involves having to take that step back and looking at the other processes in the cell and recognizing how important the energy is to driving those things. And you will soon see why it happens so quickly. So that's what that's talking about. Like I said, if you want to go and like draw out some of these so you can see what I'm talking about, think through it and see it and put it out on paper, that is a great way to get a better understanding of how these are going to be related to one another. I'm not going to ask you questions like these on the exam, of course not. But if you can understand these questions, well, you're going to be very well prepared for the exam. It's better than knowing the terms by far. So anyways, the next one, the oxygen with the carbon dioxide molecules, it didn't come from the oxygen that the aerobes breathe in. Okay, so here we're talking about when you go through glycolysis into Krebs, into EPC, right? Then at some point you're going to be giving out CO2. That's what this is starting out with, right? So it didn't come from the oxygen that we breathe in. Duh, we've said it a million times. We know that this came from the glucose from up in here, right? Not from the oxygen that went into here in order to be the final electron acceptor. But that's not what the question is asking. Okay. Let's just get into this. The question isn't asking that. That's just your setup. What role might oxygen play in how that carbon dioxide was created? So now we're asking about how oxygen, which we already said is right here, how was that involved in how this was made? This oxygen molecule, the O's are not the same O's. They can't be, it's impossible. But how are they related, right? Not just in this. This is actually asking you to take a step back out of this process. It's not asking about this right here. It's asking about what's outside of that. So this could be looked at in a number of ways, but here's one way you can look at this, just one. There are more than one. You know, go find it out, look at it yourself and decide how you feel about this, but this is my approach to how I would assess this. Last time we checked, you need O2 to go into actually, let me just step back. I don't like that. Man, that's not what I want to do. Okay. To make that CO2, which we're breathing out here, that came from glucose, right? Or something similar, but we'll just say glucose for now. 6 carbon sugar. And where did our glucose come from? It came from our food. Yeah, that's right. Okay. So where did that food come from? Ultimately. Now, if you're like, no, I can still make this work though. You can still get energy from not eating a plant. Guaranteed you're wrong about it. Guaranteed 100% you're wrong about it. You can eat meat every single day of your life. Guess where that meat came from? Guess what it had to eat? Guess where its nutrients came from? So no, it's plants every time. Plants or other photosynthetic creatures. If you want to eat algae, don't let me stop you, okay? But These guys, what they did, they took carbon dioxide out of the atmosphere, mixed it into glucose, right? So they can make other organic molecules. And for you to be able to get to this glucose, it had to build a whole dang plant. In the process of making this glucose, it also gave out this oxygen here. So in a way, the oxygen that's here did come from the same place. Not the literal same molecule, though. Because it gave you, the plants gave you the glucose that gave you the carbon dioxide. The plants gave you the oxygen that you need to breathe in so that you can get the carbon dioxide, right? This is one way to look at it. Man, I can get really complicated, and I just saw it getting really complicated for a second, and I didn't want to go that way. So we'll just stay with this here. Break it down, man. Look at it even more complicated levels, because this can be answered in a lot of different ways. Okay. This can go back to, building molecules and then getting back to those molecules and other, cycles dealing with molecules and all of that stuff. It's so much fun. It's biochemistry, y'all. Meta biochem. I don't want to do that. I'm literally just trying to pull up the pen, dude. For the last one, explain how photosynthesis powers all of your cells' functions. This is very similar to what I was just explaining previously. Photosynthesis, how does it power your cells' functions? Because you eat the plants, because your glucose and all the organic molecules that you get, because you can't use inorganic carbon, you relied on autotrophs to make it usable for you. You have to. There is no other option. So if you're relying on autotrophs and they build these organic molecules for you, and most of the autotrophs that you're going to be relying on are photoautotrophs, going through photosynthesis to get their energy, to make their parts that you're going to consume, to make their glucose that you're going to consume and use to power you and your body. That's the idea of how that will work. This is a great one to draw like a diagram, kind of like I did before. Create a cycle, if you will. Okay. I think that is everything, y'all, if you even stayed with me. I'm just checking. We got 5 people still. Okay. Do you guys have any other questions for me? Okay. If you don't, then yeah, I'm happy to do this. I know that unit 2 is difficult, and I want to give you guys as much as I can to succeed. If it helps you guys at all, then it's worth it. So yes, happy to do it. Let me know if you email me if you have questions. Otherwise, I'll see some of you guys tomorrow. I'll see the rest of you guys on Tuesday. And good luck studying. Happy studying. Email me if you have questions.